This is Pickles. Pickles is a dog. We all love dogs. And dogs love to take walks, obviously. Pickles in specific, he likes to take walks along the water. He lives in Boston, so of course he goes to the Charles River and takes a walk. Now, he goes home, takes rest, whatever it is, and in the morning, he feels off. He's not sure why, so he goes to his doctor. The doctor does some tests, and the tox screen comes back positive. Why? Well, it's because there's an algae bloom in the Charles River. Now, what is an algae bloom? Basically, when an excess amount of nutrients exist in some kind of water body, the algae is going to use those nutrients. It's going to feed off of those nutrients, and that population is going to exponentially grow. In our context, this matters because if the algae is toxic, what happens is the toxic fumes or the toxin itself is going to exacerbate in terms of its effect. And we see this not only with pickles, but across the states. In 2020, around 13 states reported around 1,200 cases of animal illnesses. Just 2 years later, that number skyrocketed to 102,000. And this is only in the US. There are headlines across the world, in China, Argentina, France, here, obviously, but headline after headline after headline shows us that this is an issue. My name is Vidyuth.
My name is Aashrita.
I'm Shaanveer.
And this is our solution to the problem.
Solutions and mitigation techniques for this issue do exist. However, many of them are inefficient, expensive, and inaccessible, such as copper sulfate treatments, which are very popular in removing algae populations, but are only short-term and actually harm the environment and aquatic life surrounding them due to the chemicals that they release into the water. On the other hand, current detection methods are extremely slow and expensive. Because of this, oftentimes, toxic algae blooms are only discovered after a large amount of civilians and pets are exposed to them, and by then, it's too late. So what can we do about this? Throughout our project, we talked with numerous stakeholders and experts in the fields of water quality and algae blooms to ensure that our project had the largest sustainable impact that it could. And through these discussions, we discovered that a project focused around developing a faster, more efficient detection method would have the largest impact, as faster detection can allow communities to be informed about algae blooms quicker, and therefore reduce the number of civilians exposed to toxic algae blooms. At the same time, the earlier toxic algae blooms are detected, the smaller they are in physical size, and the easier they are to be removed. Taking it one step further, in 2015, the United Nations identified 17 Sustainable Development Goals, otherwise known as SDGs, for the world to achieve by the year 2030. We chose to focus our project around 5 of these goals, 5 goals that directly connected to the issue that our project worked to solve. By centering our project around these goals, we made ourselves more aware about the different communities that our project could potentially affect. And it made us think more about the pros and cons of our project and the improvements that we could make to make sure that our project had the highest beneficial impact rather than a negative impact as possible. From this process, we were able to develop a detection method that doesn't require heavy machinery, doesn't require any kind of toxic chemicals, and doesn't require any materials to be released into the environment.
So what actually is our project? So we create a biosensor that predicts the production of a certain toxin called microcystin. So we looked at a specific species of algae called Microcystis aeruginosa that has 2 genes in particular that are heavily involved in this microcystin production. So we use certain technologies to detect and predict the formation of these genes. So that specific technology is CRISPR Sherlock. I'm sure many of you guys have heard of CRISPR. It's been on the news lately with gene editing. And so we use a certain offshoot of it called Sherlock that was developed at MIT. So there's 3 parts to this CRISPR Sherlock system. First, we have an enzyme or a small molecule called Cas13 that activates in a certain defined target gene that we call it. That would be those 2 genes that I just mentioned before. Next, we have a small molecule called guide RNA that essentially attaches to the Cas13 and guides it, hence the name guide RNA, to the gene that we want to look at. And then finally, we have these things called fluorescent reporters, which start glowing when Cas13 is activated. So how do we actually implement these 3 parts? We take that 3-part biosensor mix and take, ideally, an extracted RNA from a water sample. Full transparency here, because of the biosafety level of our lab and the resources that we had, we were unable to use the real water sample with live toxin. For kind of obvious reasons, we're high schoolers, we can't really deal with those toxins, but our work kind of functioned as a proof of concept for scientists of, in conglomerates or with more resources to actually do that. So we combined these two into a thing called a 96-well bioassay plate. And what we get from those target sequences and those Cas13 is that when you read it in this kind of clunky machine called a plate reader, is that you should get one group that does not glow. That would mean there's no gene in there, and that would tell us that, okay, it's safe to use, safe to drink, safe to be around. Next would be a group that did have the gene there, and that means in the future there would actually be a toxic algae bloom, then don't go there, don't swim there. And we validated these results through countless months of testing, and finally we got nice graphs to look at. So we graphed the fluorescence on the y-axis and the different groups that we did on the x-axis. And all these spikes you see, all those high bars, are positive controls, meaning there was a gene in that sample, and if it was used in a real water sample, there would be toxin. And all the smaller bars you see are negative controls. And this obviously took a lot of testing, and we ran into many errors, but finally we got nice data like this. All right, so I understand not everybody has a 96-well bioassay plate or a plate reader to actually read that on hand. So this same technology with some more resources can be adapted to this thing called an LFA. If you see on the next slide, they look kind of like a COVID test, and that's exactly what it is. You drip the water into a little test strip, and you'll get a result, positive or negative, and that'll tell you whether or not it's safe to be around, safe to walk your dog around, things like that. So if you remember from earlier, Ash mentioned that it takes up to 3 weeks currently to detect algae blooms, and by then it's just too late. The algae bloom has already produced the toxin, and it's just not safe to be around anymore. Our method takes less than 30 minutes. Let that sink in. That's 3 weeks less that you can predict the bloom, that you can be around the water, you can walk your dog, but this also goes to communities who don't have access to these large lab resources or even have the time of 3 weeks and that privilege. They get to take this little strip, test the water, and they'll know in just 30 minutes whether or not it's safe to be around.
So we just threw a lot of information at you guys, obviously. But why does this actually matter? Obviously, we are at the Pener Conference. It's about collaboration, about coming together to solve the issue at hand, climate change. And we can't do that without collaboration. And to be able to collaborate, we must help people understand these issues. If we don't help people understand these issues, there's no change that's going to happen. The practices that we have put in place, that we have normalized as a society, is what has caused this issue. And so teaching people these issues, the problems at hand, and the ways we can fix them is the only way this works. Our projects also helped work with that. We worked with children, showing them comics, up to high schoolers, and experiments with them, as well as adults. This is all part of the bigger picture, not only to find a solution to the problem, but to teach people that there is a problem in the first place, as well as a solution at hand.
We were inspired to take on this project through iGEM, an international competition that encourages high school students and college students to take on a global issue using synthetic biology. In our iGEM year, we were one of 400 different teams that took on a global issue to solve. We were even fortunate enough to meet them and understand their projects firsthand. And we discovered how amazing it truly is to be able to take on an issue and solve it with whatever skills you might have. These are people just like you and me, same age group, and honestly, personally, I didn't have any sort of experience in biology, but I still went into this, and I was still able to make a difference through iGEM, through synthetic biology, and through just wanting to make a difference.
So yeah, thank you for listening. If you have any further questions, please feel free to reach out to us. We also have a website up, so if you want to have that, feel free to reach out. But yeah, thank you so much.